Hello everyone, I am Shami Sunil Dupeska from BSc Biotechnology. Today we are going to talk on the topic Tools for DNA Manipulation, which unlocks the secret of DNA engineering. Then there are the outlines, which includes enzyme, molecular biology tools, other tools. These are the tools which we use for DNA manipulation. Then first one is enzyme, which is also act as a molecular scissor. Restriction endonuclease, cut DNA at specific recognition sites. Then there is DNA ligase, which joins two DNA fragments which were cut already. Then there is DNA polymerase, which synthesizes new DNA strands from the joined fragments. And there is reverse transcription, where we convert RNA into DNA. So the process of converting RNA into DNA includes three types of enzyme: restriction endonuclease, DNA ligase, and DNA polymerase. And then the process is empty. Then here is techniques which manipulate DNA with precision. Polymerase chain reaction. Here, PCR amplifies specific DNA sequences. Then there is gel electrophoresis, which separates DNA fragment by size. Means here you can see some small fragments which are separated by size-wise. Larger at back side and front side you can see small ones. Then there is DNA sequencing. Sequencing determines the order of nucleotides in DNA molecule. And there is gene cloning, where you create multiple copies of a same gene. So it is mostly used and important kind of technique we use. The next one is molecular biology tools, which include plasmids, vectors, primer designing software, and gene editing tools. The first one is plasmids, which include small self-replicating circular DNA molecule, which is called as plasmids. DNA molecule used to transfer genes into cells are known as vectors. You can see cell replication. Then there is primer design software, designs primer for PCR and sequence. Gene editing tools, CRISPR, Cas9, TALENS, and ZFNs. So these are the example of gene editing tools. Here are other tools supporting genetic engineering research. Microcentrifuges. This is the machine of microcentrifuge used for centrifugation and separation of DNA. You can get DNA at pellet and supernatant in liquid. Thermocycler used for PCR amplification again. Then gel documentation system used for imaging and analysis of DNA gels. Bioinformatics software used for DNA sequencing analysis and manipulation again. Here, then there is application of DNA manipulation. First is genetic engineering, to create genetically modified organism with desired trait, such as pest resistant or improved nutrition. From the gene, we remove some traits and use to produce a whole organism. Is all about genetic engineering. Then there is gene therapy, to treat genetic disorder by replacing or repairing faulty genes. You can replace it or even repair it. Then there is cancer research, to study cancer causing genes and develop targeted therapies. Then there is forensic science, to analyze DNA evidence in crime investigation. We all know it is forensic science, so this is how we use it. Then biotechnology, to develop new products such as vaccine, antibodies, and enzyme. It is mostly about medical purpose. Here comes to the conclusion. In conclusion, the tools of DNA manipulation represent a major milestone in the history of genetics, offering unparalleled opportunities for advancing our knowledge of genetics, improving human health, addressing global challenges. Thank you.